Introduction of cell division. Cell division is the process by which a parent cell divides into two or more daughter cells. Cell division usually occurs as a part of a larger cell cycle. Okay guys, today we're gonna learn about the term involved in cell division. Okay, the first term is a chromosome. This is a figure for chromosome. Okay, chromosome is a tree-like structure that located inside the nucleus of the animal and plant cell. Each chromosome is made up of protein and single molecule of deoxyribonucleic acid. Different organisms have a different number of chromosomes. For example, humans have 23 pairs of chromosomes, 22 pairs of number chromosome called autosome, and 1 pair of set X and Y. Okay, application of chromosome is in chromosome bending technique to study the primary chromosome structural changes. Okay, for the second term is a chromatin. Chromatin is a substance within the chromosome consisting of DNA and protein. The major protein in chromatin is a histone. Histone is to help package DNA in compact form that fit inside the cell nucleus. Okay, the application for chromatin. Chromatin has been used in chips or chromatin immunoprecipitation that is used to investigate a particular protein-DNA interaction. Okay, chromatids are two fiber strands which are fused together by a lone centromer. Okay, application of chromatid. Chromatid has been used in a chromatic interference in determining meiotic crossover pattern. Okay, the next term is a sister chromatid. Okay, a sister chromatid is referred to identical copies formed by DNA replication of a chromosome, with both copies joined together by a centromer. Okay, application for sister chromatid is it has been used in SCE, or we call sister chromatic exchange. SCE is a technique has been shown to have a great potential for use in a vitro screening of suspected mutagen and carcinogen.
Hi, first, what is homolog chromosome? Let's see. Homologous chromosomes are a pair of DNA molecules which contain information for the same gene, though each homologous chromosome may carry different alleles. So that is meant by homologous chromosome. The next term is tetrad chromosome. What is tetrad chromosome? Tetrad is the foursome during meiosis made by two homologous chromosomes that have each already replicated into a pair of sister chromatids. So that is tetrad. 1, 2, 3, 4. That one we call tetrad. The next up is centromere. What is centromere? The centromere is the specialized DNA sequence of a chromosome that link a pair of sister chromatids. During mitosis, spindle fibers are attached to the centromere via the kinetochore. The next term is kinetochore microtubules. What is kinetochore microtubules? Kinetochore is a protein structure that forms on a chromatid during cell division and allow it to attach to a spindle fiber on a chromosome. Centromere is where the sister chromatid connect and form a chromosome. The outer plate of the kinetochore is attached to and works in conjunction with the microtubule, which are connected to the spindles at either end of.
The spindle microtubules are protein fibers made up of as many as 45 proteins that grows from the centrioles. They form a polymer, which is a large molecule made up of many similar molecules linked together. The overall shape of the spindle is framed by three types of spindle microtubules. There are kinetochore microtubules, astral microtubule, and interpolar microtubule. Microtubules are a polarized structure containing two distinct ends, fast growing, that is plus, and slow growing, and that is minus n. Haploid and diploid cell. Haploid cells or a monoploid is a cell or organism that has just a single cell, that is, it contains one set of chromosomes. N is the haploid number. For human, N is equal to 23. These cells are formed after meiotic cell division. Haploid condition is observed in sex cells or gametes of various vertebrates. The haploid cells after meiosis are not genetically identical to their parent due to crossing over. Haploid organisms develop from unfertilized eggs. Haploid cells are important for sexual reproduction and genetic diversity. In humans, gametes, that is sperm and ova, are haploid. Haploid cells or a monoploid is a cell or organism that has just a single cell, that is, it contains one set of chromosomes. N is the haploid number. For human, N is equal to 23. These cells are formed after meiotic cell division. Haploid condition is observed in sex cells or gametes of various vertebrates. The haploid cells after meiosis are not genetically identical to their parent due to crossing over. Haploid organisms develop from unfertilized eggs. Haploid cells are important for sexual reproduction and genetic diversity. In humans, gametes, that is sperm and ova, are haploid. Somatic cells are all the cells in the body of organism except the sex cells or the gametes. All the cells except the proliferating cells of the reproductive system are somatic cells. Somatic cells are diploid with two sets of chromosomes. These cells divide by mitosis to form a new cell with identical chromosome. Out of the two sets of chromosome, one is inherited from the father while the other one comes from the mother. Examples of somatic cells are bone cells, muscle cells, nerve cells, skin cells, and blood cells.
What is gamete cell? Gamete cell are an organism reproductive cell that contain a haploid number of chromosomes. Gamete cells are produced through a type of cell division called meiosis. There is two gametes, which is male gamete are called sperm. Female gametes are called ovum. When these two gametes combine will form a zygote, is called fertilization. Application on this term is to carry out fertilization in sexually reproducible organism. What is cytokinesis? Cytokinesis is the physical process which divides the cytoplasm of parental cell into two daughter cells. Cytokinesis is one of the processes that cells have to go through to make more cells. Cells have to replicate, make more of themselves, mean reproducing. Application on this term is hair growth, nail growth, or cut on your skin start to heal. All those need more cells that requires cytokinesis. Next, karyokinesis. Karyo refers to nucleus. Kinesis is referred to division. Karyokinesis or nuclear division is the process which nucleus divided into two daughter nuclei during M phase. It can occur with or without cytokinesis, but cytokinesis is dependent on karyokinesis. In this process, the DNA condenses and the chromosomal material divides equally into two halves. What is chiasmata? Chiasmata is the X-shaped structure formed due to the point of contact or the physical link between two non-sister chromatids belonging to homologous chromosome. Chiasmata is the point where two homologous non-sister chromatids exchange genetic material during crossing over during meiosis. Finally, we will learn about crossing over or recombination. Crossing over is the exchange of segments between the non-sister chromatid of homologous chromosome in meiosis. The crossover occurs at the tetrad stage. Crossing over can be divided into three major steps, which is breakage of the chromatid segments, 2. Their transposition, 3. and fusion or joining.